So our next speaker, we're moving from spatial proteomics to spatial transcriptomics, and Matt Nilsson is also going to give a very short overview, and he's also going to talk about spatial cell mapping.
Okay. So, thank you so much. Yeah, right. So I'm going to first give a brief overview of the fundamental approaches taken to generate spatial transcriptomics data. And then I'm going to show some examples where we have used targeted in situ sequencing to make human spatial cell atlases. Right, with the probabilistic cell mapping approach. So first, conflict of interest. Co-founder of Cartana. It's a company that commercializes in situ sequencing products. But in the presentation I'm going to give later, there are no data generated with Cartana products or services. So first, general overview. So, this is not. So this is a very rapidly evolving field. It's quite exciting to see the papers coming up on bioRxiv on new methods and approaches. So the examples given here are not at all an exhaustive description of the different approaches, but I've tried to classify or identify 4 fundamental different approaches to generate spatially resolved expression data. So the first approach is to spatially barcode tissues, and that is listed here are approaches where you have spatially barcoded features on slides, to which you sort of blot over your RNA from a tissue, and then generating spatially encoded next generation sequencing libraries. So you put your libraries into a sequencer, and it will tell you where the RNA molecule came from what spatial feature on the array. It's a transcriptome-wide approach, so maybe this with the resolution is there is a lot of happening now on this in this field. So in the most recent high definition spatial transcriptomics paper, this approach was taken down to subcellular resolution to 2 micron features. But for the most, the data that has been generated out there is lower resolution, like 100 micrometer features or so. But it's scalable, and with the nanostring approach, for example, you can really draw your features, and so you could either do large features and go wider, maybe, or use smaller features and maybe have to focus a little bit. And then there are the single molecule fish based methods where you label individual mRNA molecules with cocktails of detection probes. It's a targeted approach, but in principle unlimited in multiplexing, because you do this cyclic fluorescence decoding of your fluorescence signals. You can multiplex this as far as you like. It has very high resolution. It's as good as optical microscopy gets, and a little bit better than that even. But it generates generally weak signals, so that means you have to have high-end optics and making imaging somewhat slow. And then there is where the amplified methods come into play. So you can amplify the signals from single molecule fish experiments with things like RNA-scope and HCR. And it's also targeted, obviously, but the multiplexing is harder to achieve. So currently, these approaches are limited in multiplexing. You generate stronger signals, which makes it possible to do faster imaging, and you can image, or you can map RNA in more dirty samples, like many human adult tissue sections are. And then the targeted in situ sequencing that I will be describing in more detail later, where you use padlock probes or similar approaches to target mRNAs and amplify by a Raman circle amplification. And that's targeted, and same by using the combinatorial cyclic hybridization, you can achieve basically unlimited multiplexing as well. Resolution is worse than single molecule FISH because the amplification makes the molecules bigger. Strong signals, faster imaging. And I think that human cell atlas will likely need multiple spatial resolved methods because they generate different types of data. So spatially barcoded sequencing provides shallow but untargeted information about the spatial organization across wide tissue areas. In situ sequencing also provides shallow but single-cell resolved targeted information about marker genes. And this is actually sufficient to make spatial atlases of single-cell RNA sequencing defined cell clusters across wide tissue areas. And that's what I'm going to spend the rest of my talk explaining how that works. While single molecule FISH provides very deep information about the expression states of single cells, and which can be used to refine cell type definitions and even reveal new cell types. But currently, with the heavy imaging needed, it's sort of limited to relatively small tissue areas. Right. So how do we then spatially map molecularly defined cell types using shallow targeted in situ sequencing? So this is an approach we have developed in collaboration with Kenneth Harris at UCL. And it's a probabilistic approach where we try to match the data we generate in situ with the expectations from the single-cell RNA sequencing data. It's now finally in press after 1 year of intense reviews, and it's also on bioRxiv, Qiaoyan Jian's paper. And I'll show you application of this to draw spatial cell maps in human brain and human developmental heart. So the basic approach is really like you're generating these single-cell RNA sequencing clusters, and you would like to know how they are spatially organized. So you would like to generate a map of all the cells in its its native position in the tissue. So the way we go about it is to select a set of marker genes that will, combined, be able to distinguish all these cell clusters. We make large padlock probe libraries. These are the probes we're using. These molecules become circularized when they hybridize to a specific target sequence, and then you can amplify the molecules with Royal circle amplification, generating these huge, bright blobs of DNA. And the probes are barcoded, so you basically identify the corresponding gene by a cyclic hybridization and the imaging procedure. So we generate the in situ sequencing data, so we generate molecular maps, we assign the gene reads to individual cells, and then we check the composition, the read composition of this cell, we match it to all the different clusters and see which one is the best match. And this one is clearly, we get the best match with this one. It cannot, with this combination of gene expression, we cannot be anything else than this particular cell type. So we then plot this information as a probability. It's a pie chart of a probability of match to a certain cell type. So this is what we put into our spatial maps. So the data looks like this. It's fluorescence. So here's an example cell. It's this cell sitting, this one, sitting here somewhere in the hippocampus. You generate cycles of fluorescence imaging, staining and imaging, and decode, read the barcodes, and you can assign gene identities to the barcodes. And it's really sort of high throughput. So we have so in this experiment we target 99 genes, and we targeted complete coronal sections of mouse brains, And we did actually 14 coronal sections in this experiment. And with the aim to test this approach in CA1 of the mouse hippocampus, because there a lot is known about the laminar distribution of the interneurons in this part. So we can validate and check that the cell types, the cell maps we generate actually make sense. And what's sort of extraordinary is that this can, and the maps are accurate. They really reflect what are expected in terms of the frequency of these different cell types that are quite rare, and in their location in the tissue. And this can be done with low numbers of transcript detections per cell. It's about 20, 30 transcript detections per cell, but because we're sort of relying on the deeper information from the single-cell RNA sequencing and the matching of the marker genes to the cell clusters, we can make these assignments with high accuracy. And for every cell we only record, because of the way we select the markers, we will only detect 4 or 5 or so different genes expressed in every given cell. So we also had some markers for excitatory cells in the isocortex, and when we looked into the isocortex, we could also map these. This is at the level of the cell type definitions that Nilsson published in of the whole mouse, the CNS, and it really reflects the laminar distribution of the excitatory cells and the non-laminar distribution of the interneurons, and we find the piriform cells in the piriform and so on. So it's all makes sense, and we detect very rare cells and also very common cells. So now we're sort of applying this in human cell atlas context, and we are participating in the SpaceTX consortium led by Ed Lane, which aims to accelerate the development and also comparing the different spatial transcriptomic methods and see how I can work with these kind of different data sets and also work out common data interpretation methods. And so how it works is that we have a set of single-cell RNA sequencing data. There was an agreement on marker genes that would define the different cells. So it was a joint effort to cluster the cells and then to define the marker genes. And out came a list of, say, 500 marker genes or so, and then it was up to the different spatial transcriptomic groups to select their markers for their method and generate the data and submit back to the SpaceTX consortium. And then also Alan sends out tissue to all the participating groups. So we get tissue sections from the very same specimen, so we can compare. So what we did, we targeted 119 genes in the human brain cortex MTG, so we generated data. We actually optimized the chemistry a bit, so now we generate even stronger signals, and we can decode much more dense data. So this is much more than 20, 30 reads per cell. It will be a lot more, and and we generate the molecular maps. So this is the distribution of the 190 different genes, and draw a probabilistic cell map of the 120 different cell clusters that we found on Alan Brain's homepage. Next application is in the Swedish part of the Human Cell Developmental Cell Atlas project. Here we're targeting brain, lung, and heart. And we will also do some whole embryos with time. And so we're currently focusing on generating single-cell RNA sequencing data for this. And Sten Linnarsson and his group has processed 2.5 million cells. 1.5 have passed the QC, and they have processed 480 samples from some 30 individuals. And but we're sort of the clusters are saturating, so we're sort of done with those tissues, and we're now going into the spatial step. So we're also generating spatial transcriptomic data for the tissues, and then based on the information from the single-cell RNA sequencing and the spatial data, we design padlock probe assays to do the targeted in situ sequencing. And we also have Emma Lundberg, who contributes with multiplex protein staining with the Codex platform, so we can also align protein data to the cell maps that we are generating. So as a pilot, so we haven't really, we did a pilot to see that this actually works. So we did spatial transcriptomics on 3 different ages of heart, and also generated shallow single-cell RNA sequencing from 1 heart, and developed and used this information to develop a 69 marker gene set for targeted in situ sequencing. And that is enough to resolve the 15 different cell types that were defined by the single-cell RNA sequencing. So here's the first draft map of a human developmental heart, 6.5 post-conception week. And it's sort of, yeah, it's a comprehensive spatial cell map of these cells. And you can view it on these sites. So, that's it. So thank you, the funders, and maybe particularly Chao Yen here, who generated the probabilistic approach together with Kenneth, and Marcus, and Daniel, who generated the human brain atlas. Thank you.
Thank you very much. Questions for Matt? So how easy is it to adapt this method across different tissue types, and can you do FFPE?
Yeah, yes. So it is compatible with FFPE. Efficiency typically goes down a bit because, well, it really depends on the integrity of the RNA. So it really depends on the quality of the FFPE samples.
And adapting it to different tissues, is that straightforward, or you need to do a lot of different...
So one have to work a little bit with different permeabilization and fixation conditions, typically just to optimize as any ish experiment, I would say. So what works for ish works for this.
Hello. Sorry. Thanks for your talk. And I'm quite impressed that you can use 119 genes to separate 120 cell clusters. So I'm curious how robust, how well you can separate one cell type from the other. Right.
So, you know, we get the probabilistic score for every cell. So typically what happens, so it depends on what level of, you can also look at this map on a more shallow, broader grouping of cell clusters or cell types, and then your probability of match becomes better. And then the deeper you go into the more very defined details, you often get split probabilities that this cell is either that or that cell.
So how deep you can go? For example, for Nilsson's data sets, it has 4 layers of the annotations. So which layer you can go to?
Yeah, so I can refer to the mouse because that so we have been scrutinizing this over years. I know the details pretty well there, but maybe so the deepest level we start to get so the 4th level we get split probabilities for most cells. So it's either this or that kind of situation. On the 3rd it becomes cleaner, and on 2nd it's very clean.
Okay, thank you.
One more question?
So I'm in the queue. Hi.
Oh, Sarah.
Thanks. So beautiful talk, Mats. Thank you.
Thank you.
So I have a similar question to the previous one, but basically my question is about how the ISS relates to the ST. So you were talking about how the scRNA-seq relates to the ISS and so on. So can you say something about that?
Yeah.
And how easy is it to relate them, and how informative also? Just just roughly?
Right. Yes. So we used that. So we found it useful to draw marker genes both from the single cell clusters and the spatial transcriptomics, because you get these very spatially clearly well defined distributions, which doesn't always show up in the single cell RNA sequencing data, or as one of the maybe top markers for the cells, for the cell clusters. So it adds useful information, and so I think so that's an example where I think it is really useful to have different spatial methods.
Yeah,
And also
so
add
you feel
on the
that all
proteins,
three
of course.
layers kind of complement.
Yeah,
That's what you're
they
saying.
do
Yeah.
provide complementary information.
Thanks.
Okay, great. Thank you so much for a great talk.
Thanks.